My name is Steven Reiner, and I'm with the Alan Alda Center for Communicating Science at Stony Brook University, and this is Science on Tap. I'm talking today with an extraordinary member of the Stony Brook faculty, Dr. Lilianne Mujica-Parodi, who is an associate professor of biomedical engineering at Stony Brook University. Dr. Parodi looks at the brain in extraordinary ways and is able to tell us things about how our brain operates and promises extraordinary things about how it might even operate in the future for each of us. But among the things she's done in her extraordinarily varied career is jump from an airplane at more than 13,000 feet for the sake of science. And she did this because her subjects did this also. So I want to ask you first, why did you jump out of a plane at 13,000 feet with a parachute, I might add?
Yes.
And what were other people doing?
So we were doing a study of stress resilience. And this was part of a larger effort to really think about the spectrum, the full spectrum of why some people don't do well with stress, other people do very well with stress. And what we found is that if you're operating at one end of the spectrum where you're thinking about people who are clinically anxious, it's often good enough to ask people, how do you react to normal stressors? Do you respond with more anxiety than the typical person? The problem is that when you want to look at the other end of the spectrum, when you want to look at extreme stress resilience, most people are not in a position to really compare how they would respond to an extreme stressor, because by definition it's an unusual experience.
Excuse me, so you asked yourself, well, how can I stress people out? What can I do? And who are people who do this kind of thing,
right, who
don't get stressed out, who are cool as a cucumber?
Sure. I mean, the problem with the typical stressor is that it's different for each person. Some people like horror films, other people hate them. Divorce for one person could be traumatic, and for someone else it might be a relief. And so the issue is we had to create one stressor that would be very upsetting for everyone. And ideally it would be something that would sort of tap in primally to our most basic circuits. And even more ideally, it would be something that would be translatable to animals, so that we would be able to say something in humans that then we could test in animals and vice versa.
So you had how many people jump out of a plane with a parachute?
We had several hundred people jump out of the plane as part of several different studies.
Now, without getting too specific about this, there was one entity who was very interested in your research and actually, I believe, paid for this research.
Right. So originally I was working on psychiatric research, so this was sort of more NIH type of funding.
National Institutes of Health.
National Institutes of Health type of funding mechanisms, NSF, National Science Foundation. But if you're looking at the spectrum of normal, healthy people, these foundations are less interested than in looking at disease states. And so we wound up working for many years, I would say for more than a decade, with actually the US Navy, looking at whether we could develop neurodiagnostics that eventually would be helpful in predicting whether someone would be a fantastic Navy SEAL versus be a terrible Navy SEAL.
So let's back up for a second, because you used a word we'll hear a lot about, neuro diagnostics. One word, familiar, first and second part. Neuro means brain, and diagnostics mean I'm going to diagnose.
Right.
So you want to figure out about neurological diagnosis.
So the issue that the Navy had at the time is that they would get a class of 100 individuals who'd go through the equivalent of their basic training, which is basic underwater demolition training, and only 10 to 15 out of 100 would actually make it through. And they wanted to know, is there some way that we could predict ahead of time who is likely to be able to make it through this training course versus who would not, taking into account not so much physical stamina, which is something that they were very good at screening out, but rather thinking about how individuals respond to extreme fear. So if I take you and I put you behind enemy lines, how are you going to do? And
I'm going to faint.
Well, you don't know how you're going to do, because you've never been in that situation. And so the question is, by the way, the Navy had the same problem. Until someone is actually put in that situation, how do you actually predict how someone will respond? And so what we wanted to do was to be able to put someone in a functional MRI machine. And
Let me stop you again for a second. A functional MRI machine
Yes.
is sort of an Uber MRI machine in a way. It does something different than the MRIs that we may be familiar with either personally or people we know in our families have had to have an MRI.
what you're talking about is called a structural MRI, which is looking at what the actual structure looks like.
So a snapshot
A
of
snapshot, the
static.
landscape. What we're doing is looking at the behavior of the brain, how the brain is responding to different stimuli.
So the brain in motion, in a sense.
The brain in
things
motion.
moving in the brain.
Instead of a picture, we're looking at a movie. The point was that we wanted to be able to put someone in the functional MRI, in the scanner, and probe the brain circuits and see how they respond to a very, very mild stressor, just a very, very small version of what they might be going through. And by looking at the dynamics of how the different areas of the brain were talking to one another, we would know something about how well put together that person's brain circuits were. And then that would allow us to make a prediction as to how they would respond when in a situation of extreme fear.
So the extraordinary thing is not only could you see how they're responding at that moment, but that could be predictive of how they might respond in the future because of the mathematical and other analytical tools that you threw at this.
Right, so we were explicitly and are explicitly in the prediction business. Not simply saying, this is what you are right now, but how will you evolve, how will you behave when you're put in a far different situation. And so the skydiving was developed as a way to test these methods. We would have someone go through a scanner, we'd make a prediction, they were wearing clothing embedded with biosensors, so we were measuring heart rate and respiration and endocrine measures, immune responses.
Perspiration.
Epigenetic responses, and
Epigenetic meaning
how actually your genes can change, gene expression can change.
experience can change the way your genes are expressing themselves.
Right.
What did you learn when it was all over? When all these people finished jumping?
The most extraordinary thing that we learned is that when we started, we thought that the clinically anxious people are clearly ill. These are people who have trouble getting out of bed in the morning. But we assumed that the people who were non-responsive to jumping out of a plane, these are the guys who are super cool. These are the ones who have brains that are really well put together. And in fact, it's not that simple. I mean, really, when you look at the dynamics of the circuit that we were examining, there is sort of an optimal solution to how the different parts of the brain balance the parts that are excitatory that turn the brain on versus the parts that are inhibitory that turn the circuit off.
That excite you or that inhibit you?
And that, the people who had the circuits that were best put together were actually the people who responded strongly to jumping out of a plane. Whereas at both ends of the extremes, both the people who are clinically anxious and the people who did not respond to jumping out of a plane, these were the ones who had circuits that were broken in some sense. They were broken in different ways, but it was the same circuit that was broken. And when you think about it, that's not so surprising because the purpose of the parts of the brain, the prefrontal limbic system that are meant to regulate emotion, they're there for a reason. They're to protect you from doing stupid things like jumping out of a plane. And if you are disregarding those safety signals and jumping out of a plane and not even recognizing that what you're doing is dangerous, that's a sign that your emotional circuit is not working well.
Or crossing the street when a dinosaur is coming at you quickly from the other direction, you're dead and you're not going to...
Right. So all of these, these circuits evolved within a certain context, which is, looking at predators and thinking about avoiding predation. And so they're really optimized for that kind of situation. And
So you don't want you being, let's say, military or some other entity who might want to employ skydivers. You don't want someone too brave, and you don't want someone too fearful.
Actually, so I would redefine the term brave. What you don't want is someone reckless. So what I would say is bravery means recognizing risk and dealing with it. So recognizing risk and mitigating it. I understand that what I'm doing is dangerous, and therefore I'm taking every possible precaution, and I'm going about it in a smart way. Versus people who are paralyzed by fear and are not able to respond, which is maladaptive, or people who do not recognize fear, in which case they don't take steps to mitigate risk, which is also maladaptive.
Now, along the way, and correct me if I'm wrong because I'm not sure I have the sequence right, but along the way, you discovered something else about how the way that people, human beings, actually can communicate their fear to one another in a very nonverbal way. Tell us about that.
Well, at the time as I mentioned, I was putting people in the scanner, and I needed to get the part of the brain activated that was responding to fear. So the question is, how do you make a human being a little bit afraid? And that's not so easy to do. I mean, the standard ways that people do it in my field would be, you show someone an angry face, you show someone a horror film, but we're smart, right? We know that an angry face is not really a threat. We know that a horror film is just a bunch of actors. And so I became interested in seeing whether we could actually tap into a very primitive mechanism that other animals use to communicate fear. And those are called alarm pheromones. So fish have alarm pheromones, insects have alarm pheromones, and even mammals have alarm pheromones. What are
You use an example of deer. We've all maybe had that experience.
So you see one deer, one of them sees you and all of a sudden they all run. How do they communicate? This is kind of one of the important factors in flock behavior. And the way this is working is that one animal that detects a predator emits a gas that actually is detected unconsciously by others of the same species. And if you think about it, this is a very elegant solution to the problem of how to communicate that there's a predator around. Because certainly if you're a little mouse and you're in a field and you see a cat, screaming and running, jumping up and down is not a very adaptive response. Because certainly your colleagues will know that there's a predator around, but you will also die. So, so the ability to actually covertly send a signal is actually very useful. And so I thought, well, this had never been shown before, but there had been some talk over the last 10, 15 years that humans had reproductive pheromones. And I thought, well, if all other animals have both reproductive and alarm pheromones, and we have reproductive pheromones, perhaps we have alarm pheromones as well. And since I was in the business of scaring people on a regular basis anyway, I thought, well, why not capitalize on that?
It's actually interesting we're doing this just a couple of days after Halloween. I just thought of it.
And so we wanted to actually capture the sweat and compare it. So we wanted to make sure, for example, that whatever response people were getting to a sweat was not just due to an odor. So we collected sweat under essentially sterile conditions because it's a bacteria that gives sweat its bad odor. So we had the same individuals provide the stress sweat and also control sweat. So on one day they had an emotional but not physical stressor, which was jumping out of the plane, and on the other day they had a physical but not emotional stressor, which was running on a treadmill for the same period of time at the same time of day. We took the sweat under both conditions, extracted it, pulled it, and then presented it via what's known as an olfactometer to a second group of individuals that were undergoing fMRI. So this is allowing them to breathe it in. Now the reason this was kind of an elegant design is that it was really a double-blind, placebo-controlled study. We had individuals in the scanner who did not know what they were breathing in. It had no odor, and there was nothing about it that would make them think that they were breathing in the sweat of people who were frightened. All they knew was that they were breathing. So they were breathing in, breathing out, breathing in, breathing out. And what was quite extraordinary was that the fear center of the brain, the amygdala, responded selectively to the fear sweat, but not to the exercise sweat. So it showed quite conclusively that humans were able to detect fear in other humans without conscious perception.
So if I'm sitting here right now and you're scaring me to death, and I have a lot of anxiety because I'm in front of some individuals, and there's cameras here, and I'm perspiring profusely, but you can't really see it, or thank God, smell it. I am still communicating something to you. What's it doing to you?
Well, you might think that what it's doing is scaring me or scaring other people.
Or I'm giving off bad vibes and I'm getting you stressed out somehow by
Well,
my
I was
stress.
the one who stressed you,
Right,
so
you
that
stress
doesn't
me.
make sense.
Right.
But maybe you're
sensing my
stressing everybody else.
Right, maybe I'm stressing all you guys out.
Okay, so it doesn't exactly work that way. I mean, and again, it comes down to this whole idea of how is this an adaptive mechanism? So again, we have the little field mouse. The field mouse becomes frightened because it sees the cat. And so he sends out this covert signal that there's a predator around, there's alarm. So what happens if now all the little other field mice start freaking out? Freaking out is never an adaptive response under any circumstances. And so what happens is that the other field mice, it's not that they freak out, but they freeze, so they stop, they freeze, scan, and sniff. And that pattern of freezing, scanning, and sniffing allows them to gather more information and allows them to then detect the predator, identify what type of predator it is, and then get away. And what we found is that it does something very similar in humans. It's not that it frightens them per se. It puts them on high alert, and it allows them to pay attention to cues that otherwise they wouldn't pay attention to.
So it even sharpens their sensibility.
Yes, it sharpens their perception. So people become more accurate in detecting threat. So if there is an actual threat, they become better at detecting it. And if there isn't a threat, they also become better at detecting that there isn't a threat.
So it's actually, there is an adaptive strategy, and it's good for the sake of the group to have someone or someone's actually reacting in a fashion that alerts others without waving your hands and jumping up and down, which will get
Right. So I mean, most of the time we're not on high alert. Most of the time we're, grazing and eating little berries or whatever. But some of the time, we can't be on high alert all the time. And so when you get the signal, it causes you to stop and pay attention and to gather additional information.
Now, not that any of this is not very serious and important with lots of applications and implications, but it's not right now the central focus of what your research is. And I think to get into that, we have to understand a little bit about how the brain attempts to stay in balance and how finely tuned the brain really is. And because we get from stress and anxiety to really troubling psychiatric problems like schizophrenia and depression and bipolarity and other psychiatric ills. So I want you to, if you can, just maybe walk us into that field, because ultimately what you're talking about, I believe, when you're talking about personalized neurodiagnostics, is to be able to look into my brain and, based upon giving my brain certain stimuli, be able to predict with a certain certainty whether or not I may be vulnerable to certain neurological diseases.
Right. So this was always the intention, and the stress studies were a way to develop methods. They were a way to take individuals across the spectrum that we knew that the same circuit was involved, and we wanted to know if we could actually predict where along that spectrum people would fall. Because ultimately we wanted to bring this back to thinking about neurological and psychiatric illness. And again, we're in a prediction business, right? So certainly it would be very useful if we could take someone who comes into a psych ER, put them in a scanner, figure out exactly what the diagnosis is, what medication they ought to take, and what the dosage is. But ultimately, we're trying to do something much more ambitious, which is not to take people who are currently symptomatic, but rather to take individuals who are not currently symptomatic and to predict the risk for them developing a future illness. So this could be likened by analogy to what a glucose tolerance test would do if in fact you thought you were at risk for developing diabetes. So let's say that you have diabetes that runs in your family, or you're in a state like pregnancy that predisposes you to diabetes. So your doctor would take you in and give you a bolus of glucose and then watch how your system then regulates as it returns back to baseline. And that would tell your doctor something about your glucose and insulin regulation. Again, thinking about parts of the circuit that turn on versus turn off and how they're balanced with one another in returning back to baseline and homeostasis. And what we're trying to do is something very similar, to take individuals who might be at risk for developing neurological or psychiatric illness and to be able to look at those inner workings, the dynamics of that particular circuit, and to be able to, on the basis of that, to make a prediction of how the system will evolve over time.
Now, when you say might be at risk, these are individuals who have a family history or?
For example. So in some cases, they would have a family history, but you look at some types of illnesses like Alzheimer's disease or dementia, we're all at risk for these. So even just having a general sense of what path you're headed towards would be enormously useful in terms of thinking about developing treatments and knowing when to administer them.
Now, just backing up for a second, I thought it was interesting when you pointed out what we kind of all know, in terms of right now, in terms of psychiatry and diagnoses, how do we diagnose psychiatric problems and illnesses, and how do we go to try to find remedies? How inexact is it?
Right. Well, at this point, psychiatric illnesses are diagnosed literally by going in front of a doctor who asks you questions and you answer how you feel. And that has a number of limitations. One is that it depends on the patient being honest about the signs and symptoms. In many cases, there are good reasons not to be honest. So if you know that someone might lock you up on a locked ward, if you say that you're hallucinating, you might not say that you're hallucinating. But in some cases, people are not in a position to report on their signs and symptoms. So for example, if you have an individual who's been depressed for the last 20 years, that individual might not have an adequate frame of reference for saying I'm depressed, because that person might not recognize what it is not to be depressed. And so what we'd like to do is to bring psychiatry and neurology, and this is less of an issue with neurology, but it's still also an issue to some degree depending on the illness, more in line with, say, cardiology or endocrinology, where if you have some degree of risk or you're having troublesome signs and symptoms, you can have a blood test or you can have an EKG. And on the basis of the blood test or the EKG, there's a certain range of what is normal, and either you're too high or too low, but at least you have then an objective benchmark for knowing what is going on and how to treat it.
Well, this seems potential to shake up the status quo in this entire field of medicine, to shake up the status quo in the pharmaceutical industry.
Yeah, I mean, I think ultimately everyone benefits by this, because from the point of view of the pharmaceutical companies, they can then develop drugs that target a certain portion of the population. It's really clear who's going to be benefiting from them or not. Their clinical trials will be more successful because they won't be mixing in people who are likely to respond with people who are not likely to respond. Certainly, patients would benefit in that they wouldn't have to go through a regimen of trying many different drugs to figure out which one is working best or figuring out what is the appropriate dosage. This is not a unique idea on my part. I mean, certainly biomarkers are something that neuroimaging has been looking for a long time. I think what makes my research unique is that my conception of what a biomarker is quite different. Instead of saying that it's a snapshot of an area of the brain that is too high or too low, it's on too much or it's off too much, or talking about whether two brain regions are more connected or less connected in this population versus the other, I'm really looking at a negative feedback loop and the dynamics of a negative feedback loop.
Could you define that for us, what a negative feedback loop is?
Well, I can give an analogy that might be familiar to everyone. So I mentioned before that the body needs to maintain itself in a state of homeostasis, right? So we need to have lots of different variables that are within certain tight limits. We have to have our temperature within a certain specific range. We need to have our acidity level within a certain specific range, our salt levels, and so forth. And so the body has mechanisms for pushing you up or pushing you down depending upon where you lie along that range. And we can say, let's say we want to maintain a particular temperature in this room. We have, things called heaters and air conditioning that could be likened to a heater would be an excitatory influence, which is pushing the temperature up. The air conditioning is an inhibitory influence, pushing the temperature down. And certainly what you need is also an appropriate sensor. You need something that is telling the system, have I passed this threshold or not, and how do I respond?
Every thermostat has that little thing of mercury or whatever it's made out of.
Now, the way in which we differ from a thermostat is that the thermostat needs to be only at one particular place. And to a certain extent, that's true of our glucose and acidity levels and salt levels and so forth. But in the case of our brain circuits, in most cases, we don't want to be stuck just in one place. We want to be in a state not of homeostasis, but what's known as allostasis. We want to be able to respond to the environment and then also return back to baseline.
You want to be scared and then get calm.
Right, and you need to be able to be scared,
But you need to be able to
and you need to be able to return. So in a way, the system has to be very delicately balanced. It needs to be balanced in such a way that it really requires a minimum of energy to make the system respond, but then also return to baseline.
Well, is this a theory? Is this a question of, in other words, from where we are right now to where you want to go in your research, to getting this so it's, this is what is, this is what's done. Mm-hmm. What has to be accomplished? You need money, you need grants, you need support. Do you need a breakthrough technologically? Do you need simply more time to do your experiments? What is between now and where you want to be?
I mean, one of the things that hampered us the most originally was that we were relying upon traditional fMRI, which didn't have signal that was strong enough to be able to look at individual time series. So we could
individual
time series. So we're looking at a response that occurs over time.
And what period of time? It could be from, what's the range?
Over an hour, let's say.
Okay.
Over the course of an hour. And the problem is that, what we're looking for in order to derive these systems, so what we're doing is creating a computational model for each person's brain, but in order to do that, we need something that looks like this, and instead we're getting something that looks like this. So it's a big mess. And the way in fMRI you deal with this big mess is that you combine lots and lots of trials. So you do something over and over and over again, and you combine the trials. And then on top of that, you combine lots and lots of people. And the idea is that the signal will amplify, whereas the noise, this part that you're not interested in, will cancel out. The only problem is that if I want to develop a neurodiagnostic, I cannot rely upon 30 people in one group and 30 people in another. If you come to me and say, am I going to develop dementia? You don't want me to put you in a group of 30 with another group of 30 and say, okay, well, your group will develop, something, but I can't say anything about you. You want to know about you. And so the problem was that we needed a way to amplify the signal, and that's when we started looking at actually moving to ultra-high field neuroimaging.
It's just more and more powerful.
Right. So yes, we get a better signal. And ultimately, what we're trying to do is to focus again on identifying this very, very small, very subtle dysregulation. Again, that's why the signal needs to be so strong, because we're not taking someone who shows frank illness. We're taking someone who shows a very, very small, mild dysregulation that over time then will become more and more severe and eventually will become symptomatic.
So take us for a second, and then we'll open this up to some questions if that's about time. Take us for a second, 5, 10, 15 years into the future. Your research, your investigations have been a tremendous success. You've managed to accomplish what you wanted to accomplish. You've been proven right. You got a lot of money from all sorts of funders.
So this is some sort of fantasy, right?
Right, some sort of fantasy. How are our lives different? What do we know about ourselves that we might not have otherwise known, and how might that help us live better lives?
I think the number one thing that would change is the ability to make real what people have been talking about in the form of personalized medicine, right? So everyone is different, And again, most of research has really been focusing on groups. But ultimately, what we want to be able to do is to take a single individual. And in our case, what we're trying to do is to create a situation where we can have one individual, we take a brain scan, and we come up with what is essentially a computational model of that person's brain. So your brain, or your brain, or your brain. And then the advantage of having that model is that we can then put in a new input. Not your current situation, but what would happen if we did this. And that input might be, for example, a drug. It might be putting in a new drug. And then ask, now how will that model, what will be the trajectory that it will follow over time? How will the evolution of this trajectory occur? And that will then, in principle, allow us to be able to predict how someone responds, not just during the scan, but months from then, or years from then, or decades from then. It should be able to predict whether someone is going to recover, how quickly they'll recover, how quickly they'll degenerate, whether the process will be an oscillation. So there are certain types of mental illness that are oscillatory.
Change up and down.
Like bipolar disorder, for example.
Back and forth, back and forth. Right.
So, I mean, some people with bipolar disorder cycle very quickly, some people cycle more slowly. The question is why, and this is something that we're hoping will allow us to answer that.
And so basically improve our lives, because it'll give us more information, or does it give us more to be scared about, more to be frightened about, more to feel out of control about? Maybe people say, I don't want to know if I'm going to get Alzheimer's 20 years from now, or if I'm likely to get it, because...
Then don't have the test.
Then don't have the test.
Don't have the test. I mean, that's equally true for every other part of medicine. You don't have to have an EKG. You don't have to have a blood test. But, again, we were talking about what is it to be brave. I mean, to be brave is to recognize risk and take rational steps to mitigate it. And to me, this is part of being brave. It's recognizing that we are mortal, that we're susceptible to certain diseases, and dealing with it, getting the information we need so we can appropriately treat it.
Okay, I think that's a very good note to end on with all these brave people in the audience. So thank me very much. Thank you, Dr. Mujica-Parodi. And do we have any questions? We have a microphone. Would anyone like to get scanned, because we've got one outside?
Were there any patterns, men versus women, age, education, background, occupation, anything that gave you a hint in advance, or when you looked at these populations about who could jump out of the plane, or who could find that balance and who couldn't, who were on the extremes? Anything. What
was really extraordinary is that when we had people jumping out of planes, we also did a whole series of clinical assessments. We had psychiatrists and psychologists who asked these individuals all the typical questions you would expect someone to ask. And of course, this was part of our mission in trying to develop neurodiagnostics for the SEALs, because they have the SEALs psychologists who try to identify ahead of time who's likely to succeed and who wouldn't. So they asked obvious things like, how do you deal with stress? Did you grow up with a lot of stress? How did you deal with it when you grew up? What are your parents like? That sort of thing. It turns out that not a single clinical measure, not a single questionnaire subjective type measure was predictive at all of how people responded, not even slightly. What was predictive were biological measures, and the two that were most predictive, well, primarily was the brain. The brain was what was most predictive, and to a secondary degree was age.
And what did you find about age? The older you get, the braver you are, yes? No, that's not true? That's not true?
That's why I said this is a brave group.
That's not true?
So the older you are, the better you are at assessing risk. And from that perspective, because that is how I would define bravery, that is true. That as you get older, you do become braver. And so what happens is that you have younger individuals who I would say tend to either be very fearful of what they don't know because they've had very limited experience. So it is much more frightening because they've had much less exposure. Or at the other extreme, you have people who are very reckless.
And with that, I think we want to thank this brave group. Wants to thank you very much for a fascinating evening. So thank you. we'll have another one of these Science on Tap programs coming up at the end of this month, and this will be posted on the Stony Brook Aldous Center website. So thank you very much for coming, and we'll see you again.
Thank you.